The overall goal of this procedure is to simplify readout of digital droplet assays using a conventional real-time PCR instrument to measure bulk fluorescence of the assay. This is accomplished by first preparing amplification reactions of the samples of interest in two standards, and forming droplets to partition each into thousands of nanoliter-sized reactors. The second step is to incubate the droplets to amplify the nucleic acids within each droplet. Next, the bulk fluorescence of each sample or standard is measured with a standard qPCR thermocycler. The final step is to predict the fraction of fluorescent droplets in the samples of interest using a standard curve generated from the standard samples. To verify the performance of this method, fluorescence microscopy can be used to independently evaluate the quantification performance of the bulk fluorescence assay.
The main advantage of this technique over existing methods, like droplet counting, is that it does not require specialized instrumentation.
Prior to starting this assay, prepare all necessary reagents as described in the protocol text. Two standards are required. One no template control, such as nuclease-free water, and one high template concentration, such as lambda DNA. Denature 3.3 microliters of each standard with 3.3 microliters of denaturation buffer in a qPCR tube. Incubate at room temperature for 3 minutes. Quench each with 3.3 microliters of neutralization buffer. Similarly, denature 3.3 microliters of the template of interest with 3.3 microliters of denaturation buffer. Incubate at room temperature for 3 minutes. Quench with 3.3 microliters of neutralization buffer. Prepare 11 microliters of master mixture per sample. For fewer false positives, expose the master mix to UV light prior to forming droplets. First